Assalamu alaikum. This is the presentation of the endocrine data show. First, this is the pituitary gland. We all know that it has a posterior lobe and an anterior lobe. The anterior lobe is forming a C-shaped structure, and in the concavity of the C we have the posterior lobe. Here we have the pars intermedia and then the hypophyseal cleft, which is this white space representing the remnants of the lumen of the Rask pouch from which the anterior pituitary arises, and this is the pars distalis of the anterior pituitary. So this dark area, it is the pars distalis, and this is the hypophyseal cleft, and this is again a dark area which is the pars intermedia, and then it is joining the posterior lobe, which is faintly stained or pale stained. Here we have a transverse section, but here we have a longitudinal section so that you can see the pale area is the pars nervosa, and here we have the connection with the hypothalamus, which is called the infundibulum. And here we have the dark area representing the anterior pituitary, and this is the pars distalis. Here we have a cleft here, and then the dark area here, it is the pars intermedia, and the upward prolongation of the pars distalis is the pars tuberalis. Here again, the lightly stained area is the pars nervosa, and this is the pars intermedia on the surface of the nervosa immediately, and this is the gap, which is the hypophyseal cleft, and this is the pars distalis. Again, the pale is the pars nervosa, showing the fenestrated blood capillaries, showing the nuclei of the pituitarides, and showing these red fibers, which are the unmyelinated axons of the hypothalamo-hypophyseal tracts, which are the axons of the neurosecretory cells high up in the hypothalamus. And then this is the pars intermedia formed of basophilic cells, as you see. It is darkly stained compared to the pars nervosa, and the cleft, the hypophyseal cleft, and then you will move to the pars distalis, the biggest part, containing acidophils, containing basophils, and also unstained areas of chromophobes. Here, chromophobes which are not stained. Here, a chromophobes, typical chromophore. The nucleus is stained, but the cell cytoplasm is not stained. Here again, this is the number 1 here is demonstrating the acidophils. Number 2 here, it is demonstrating the basophils. Number 3, the chromophobes, which are not stained, and also this is a chromophobe. Then number 4, we have the pars intermedia basophilic cells. Number 5, we have the fenestrated capillaries to allow the rapid entry of the hormones. Number 6, it is the pituitarides nuclei. Number 7, the non-myelinated, this red area, non-myelinated nerve fibers, which are the axons of neurosecretory cells in the hypothalamus. Here we have a high magnification for the pars distalis, where we can see the chromophobes forming 59% of the total cells of the pars distalis, and the acidophil, which is one of the types of the chromophils, and they are forming 37%, and the basophils forming 11%. Both the acidophil and basophil are the chromophils, which are 48%. Here we have the posterior pituitary showing 4 structures. We have the red fibers, which are the axons of neurosecretory cells, supraoptic paraventricular nuclei present in the hypothalamus. And here we have the nuclei of the pituitarides, and here we have the fenestrated capillaries present here. Of course, we have herring bodies, but they are not present in this part of the section. Here we can see the herring body, which is acidophilic, pale stained, and it is not provided with nuclei like those of the fenestrated capillaries here. And the fenestrated capillaries are more darkly stained, containing blood, but here it is lightly stained, no nuclei, and these are the accumulated secretions herring bodies. Here we have the nuclei of the pituitary, which are star-shaped cells, but we can see only evidently the nuclei and some of the extensions of the cells. And here we have the non-myelinated nerve fibers. So in this picture you can see the four structures: fenestrated blood capillaries, unmyelinated nerve fibers, hailing bodies, pituitary. The second gland is the suprarenal gland. The suprarenal gland is found above the kidney, and it is formed of a capsule, and under the capsule we have a cortex, peripheral part, and medulla, inner part. Here the cortex is divided into three zones according to the arrangement of the cells. In the superficial part we have a narrow zone in which the cells are arranged in the form of curved arches, looking like glomeruli, means rounded structures, so this is called zona glomerulosa. Then the cells here are arranged in straight fascicles. Fascicles means narrow straight rows here, and here we have it is wide area, and the cells are appearing to be pale compared to the others due to the presence of a large amount of the fat droplets which disappear during preparation of the section, dissolve. So this is the pale area, it is the zona fasciculata. Fascicle means حزمة بالعربي, in Arabic يعني this is the area in which the cells are arranged in straight rows beside each other, and in between we have the fenestrated blood capillaries. Then we have the red area appear again, in which we have the cells in the form of a reticulum, and in the openings of the reticulum you can see the blood sinusoids. Here we have blood sinusoids, here we have fenestrated capillaries, here we have fenestrated capillaries, but here we have blood sinusoid. Then we will move to the medulla, formed of chromaffin cells, which are arranged also in the form of a reticulum, and they having the blood sinusoids. Also, this is a very low magnification. You can see the capsule. Then we have, this is the cortex, and this is the medulla. In the deep part of the cortex, red area means the zona reticularis, the light area means zona fasciculata, and the relatively darker here, it is the zona glomerulosa, and this is the capsule. Here again we have the capsule covered by fat cells, as you see, and including also some fat cells. And here we have the curved arches of the zona glomerulosa, formed of columnar cells with basal rounded nuclei secreting the mineralocorticoid aldosterone. And here we have the lightly stained area, which is the zona fasciculata, formed of this pale stained area due to dissolving of the lipid droplets, giving them the name spongiocytes. And in between we have the fenestrated blood capillaries. Also we have here fenestrated blood capillaries in between these arches. Then we will move to the red area, which is formed of a reticulum of the cells, and in between you can see spaces like this one, which is blood sinusoids. And then in the deeper part you can see the start of the appearance of the medulla, containing also blood sinusoids. So again, this is the capsule, this is the glomerulosa, and this is the fasciculata here. Here we have fenestrated capillaries, here we have fenestrated capillaries. Then here we have also the capsule, very excellent picture for the zona glomerulosa, curved arches, and in between we have the fenestrated curved blood capillaries. And then we have the zona fasciculata, pale stained, and then the dark appearance here demonstrates or denotes the appearance of the zona reticularis, and in the deep part we will reach the medulla, which is not clearly seen here in this picture. Here again we have the zona fasciculata, characterized by the presence of these multiple fat drops which dissolve, giving the appearance of a sponge for the cell, so they have another name, spongiocytes. Here we have the zona reticularis, formed of cells forming a reticulum, and here we have the cells with the evident acidophilic cytoplasm, not vacuolated like this one, so the color here becomes darker. In between these irregular branching course of cells, we have these white spaces demonstrating the blood sinusoids. And here we have the medulla, the same appearance as regards it is a reticulum, but here the cells are basophilic. Notice the evident nucleus. Nucleolus means active cells, and these are the chromaffin cells, and these spaces are the blood sinusoids. Here we have a picture for another gland, or actually two glands beside each other, the thyroid and the parathyroid. Here we have the follicles of the thyroid full of the colloid, and the wall is formed of cells having rounded nuclei. These are the follicular cells. And here we have another gland formed of mainly dark cells together with blood vessels or blood vessels and fenestrated capillaries. So on the right side, this is the thyroid. On the left, it is the parathyroid. The thyroid is characterized by the pink colloid secretion present inside the follicles. Here it is somewhat destroyed by the fixation process that cause the separation of the secretion from the wall. But still you can see the rounded nuclei of the lining cubical cells of the thyroid follicles. Also, these are blood vessels. And here we have the parathyroids. The cells are small, forming a huge collection of cells not in the form of follicles, and mainly they are basophilic. Here, by higher magnification, you can see clearly the cubical cells, evident cubical cells with central rounded nuclei. And here, these shrunken colloid, and this shrinkage takes place during the preparation of the section as an artifact. It is not normal. It is not the normal picture of it, but it is due to the preparation of the section. Here we can see also cells which are here clear, or it is of pale cytoplasm. They are called C cells secreting the thyrocalcitonin. Here is the colloid, and this is the follicular cells, and this one here, it is the C cells or the parafollicular cell. On the right side, you can see the fenestrated capillaries, very evident here, and we have huge collection of cells which are basophil cells, which are called chief cells. Here we have the higher magnification, the thyroid follicle formed of the principal follicular cells, cubical cells with central rounded nuclei. And in addition to that, you can see cells which cannot reach the lumen. They are called the parafollicular cells, which are pale stained, and they are called C cells or parafollicular cells secreting thyrocalcitonin, while these cells are secreting the thyroid hormones T3 and T4 into this colloid. The colloid here is pink, and it appears retracted after the fixation process, as we have said before. Then we have here the parathyroid. Here we can see that the majority of the cells are darkly stained basophils called chief cells. Here we have the fenestrated capillaries. Here we have the fenestrated capillaries. And here we can see a lesser number of cells of acidophils. They are called oxyphil cells, which are acidophilic in staining. Here again we have the basophils, which forming the majority, so they are called chief cells. They are small in size but more in number. Here we can see the fenestrated capillaries in this area lined by the flat squamous cells, fenestrated capillaries, and here you can see these larger cells but lesser in number called oxyphil cell. Take care. Don't mistake these cells for oxantic cells of the stomach. This is oxyphil cell, which is secrete. It is of unknown function till now, and it increases by the advance of age. But these cells, which are the chief cells, secrete the paras hormone, which increases the blood calcium level. Trial exam number 1. Identify this organ. This organ is the suprarenal gland, because I can see these glomeruli and I can see the pale fascicles. Identify the region indicated by the arrow 10. It is the zona glomerulosa of the suprarenal. Identify the region 11. It is the zona fasciculata of the suprarenal gland. What does this area produce? They produce the glucocorticoids. Here, the second, it is the thyroid gland. Identify the entire structure, it is the thyroid follicle. Identify the material indicated by the arrow. Here it is a colloid, which is containing the stored hormones. Identify, number 4, the granules. Here, these granules are present in the follicular cells. Actually, these granules appear very clearly by the electron microscope, as you can see that in your textbook. But here you can see, by this high magnification, you can see these accumulated secretory granules. So 4, it is the secretory granules here in the follicular cells. Identify this organ. This organ is showing the basophils, acidophils, blood capillaries, and cells which are not stained. However, if you are not sure about the diagnosis, try to read the rest of the questions. The rest of the question will help you to diagnose what is this like, because he is asking about percentages. So, this organ is a pituitary gland, and what region exactly? It is the pars distalis of the pituitary gland, but you should say the organ is pituitary gland. Between two brackets, you can say the pars distalis, or you cannot say that at all because you are asked for the organ itself. The black arrow points to the basophils. Their percentage is 11%. The yellow arrow points to acidophils. Their percentage is 37%. The green arrow points to fenestrated blood capillary. Number 4, this is a section of the parathyroid. Identify the cells by the black arrow. It is the oxyphil cells. These cells increase or appear after some time of your age, not from the start, and it increased with the advance of age, of unknown function, and they are acidophilic, and they contain by the electron microscope large number of mitochondria. Here we have the chief cells, which are the basophils, indicated by the yellow arrow. These are the chief cells which are basophilic, and they are the cells responsible for the production of the parathyroid hormone that reduce the blood calcium level. Here, this is section in the pars nervosa of the pituitary gland. The black arrows points to fenestrated blood capillaries. The blue arrow points to here the pituitary. The star here denotes the axons of the neurosecretory cells, or unmyelinated fibers of neurosecretory cells. Number 6, identify the organ. This is, of course, the suprarenal gland. The red arrow points to the zona glomerulosa, while the green arrow points to the capsule. The blue arrow points to here, the blue arrow points to here, it is the start of, it is the area of the zona fasciculata, because we have these spongiocytes which secrete glucocorticoids. The yellow arrow points to here, the yellow arrow here points to the area deep to the fasciculata, darker than the fasciculata, so this is the zona reticularis. And the orange arrow points to here, it is present in the area of the fasciculata, so it is the fenestrated blood capillaries. Number 7, identify the region of the organ. So it is the pars nervosa, and identify the structure. This is number 17, it is the pars nervosa. Number 18, identify the structures indicated by the arrow. It is the pars intermedia. Number 19, identify the cells indicated by the arrows. These are the pituitary. Number 20, here it is the pars intermedia. Again, identify the region of the organ. It is the number 17, pars nervosa. 18, here it is the 18, this is the fenestrated blood capillaries. Number 19, pituitary. Number 20, it is the pars intermedia of pituitary gland. Here, identify the organ. It is the thyroid gland. The material indicated by the letter A, it is the colloid. The yellow arrow points to the parafollicular cells, and the blue arrow points to follicular cell. And of course, you should know their functions. Number 9, identify the part of the pituitary gland, pars nervosa. The black arrows points to, here black arrows points to the pituitary, while the curved arrow points to fenestrated blood capillary. The blue arrow points to herring body, and the star points to nerve fibers or unmyelinated nerve fibers of neurosecretory cells.